Well, thank you so much, everybody, for being here. I very much appreciate that kind introduction, and also I'd like to thank Dr. Jonathan Su for the invitation to speak to you tonight. And so I've been asked to talk about our lab's role in the development of prevention strategies for HIV infection. So I'd like to introduce you to my laboratory. This is the Clinical Pharmacology and Analytical Chemistry Lab at UNC. It's comprised of a wonderful multidisciplinary group of individuals. We have clinicians, we have pharmacologists, chemists, engineers, mathematicians, and a wonderful group of PhD students and postdocs. We are a global lab. We work with multiple nations through funding and through partnerships. We also work closely with the NIH, with the World Health Organization, with the Centers for Disease Control. We work within Cura, which is the first public-private partnership at UNC focused on curing HIV. We've worked with a lot of pharmaceutical industry, and most recently we're working with the Bill and Melinda Gates Foundation. We are a certified lab. We are CAP and CLIA certified because we do provide a lot of data to the Food and Drug Administration, the European Medicines Agency for support of investigational new drugs or new drug application submissions. And we are also a core laboratory where we work in the preclinical and clinical pharmacology space for the UNC Center for AIDS Research. So in my laboratory, we are both experimentalists and clinical trialists, and the goal of clinical pharmacology is really to get the right drug to the right site at the right concentration for the right amount of time. So we work in cell models, both static and dynamic cell systems. We work with tissue models. We work in animal models, primarily non-human primates, and also humanized mice, so mice that have a human immune system. We do a lot of mathematical modeling for prediction of drug concentrations and drug dosing. And we also conduct clinical studies in healthy volunteers or in HIV-infected volunteers. We have a focus of the laboratory on treatment, particularly in special populations like pregnancy. We've done a lot of work in HIV prevention that I'll talk to you about tonight, and also in the eradication space. So one of the tools that we use in clinical pharmacology is really to define the pharmacokinetic and pharmacodynamic relationships of a drug. And so pharmacokinetics can really be defined as what the body does to a drug, how it absorbs a drug, how it eliminates a drug. And then pharmacodynamics is what the drug does to the body, either in terms of efficacy or toxicity. And so kinetic and dynamic relationships are really important to design rational drug dosing, frequency of dosing, drug combinations, and duration of therapy. And so in the drug development process, it goes something like this. We give a dose of a drug to a person, and then we get a concentration within the body that's circulating through the systemic circulation in the blood or in the plasma. That drug then finds its way to the effect site, and for HIV, it's in tissues or in cells. And then we have some sort of a pharmacologic effect, in this case, impact on viral dynamics. So pharmacokinetics typically encompasses the left-hand side of this slide, so between the drug dose and the effect site concentration, where we can define concentration-time relationships. And then pharmacodynamics is typically between the effect site concentration and the effect that you're trying to achieve. So this is more of an effect versus concentration relationship. And with these two things, we can come up with how to optimize dosing strategies in people. However, the effect site concentration, that tissue or that cell, is often hard to come by. It's very hard to sample. It's challenging to do in clinical studies. And so most often what we see is pharmacokinetics done in plasma or in blood, where we can take blood samples and we can measure the concentration of the drug over time. And then we make some assumptions about what that concentration in plasma actually does for an effect. But the problem is, particularly for HIV, because HIV is found everywhere in the body, that tissue drug distribution is quite heterogeneous. It's not homogeneous, and it's tissue specific. So you can have high intra-tissue variability, inter-tissue variability, and inter-subject variability. So this figure is a PET image of an antibiotic called trovafloxacin. And so high concentrations here are in pink and in orange. Lower concentrations are in blue and in dark purple. And what you can see is whether you're looking at the liver or you're looking at the heart or the bowel or even the kidney, that there are different distribution patterns of drug, both within an organ and between organs. So then having one plasma pharmacokinetic profile to try and predict what's going on in different tissues is really hard. So for example, if this is your concentration time profile in plasma after you've taken a dose of a drug, this may be your concentration in spleen, and so that's pretty close. But this may be your concentration in brain, and then this may be your concentration in gut. So trying to just look at plasma and make assumptions about what's going on in different tissues can be really hard. So why is tissue distribution heterogeneous? Well, there's a number of reasons. There's physical chemical properties of the drug can cause different exposures in different tissues. Plasma protein binding can change the way a drug is distributed in different areas of the body. If the drug acts intracellularly, like most of our drugs for HIV need to act, if there's conversion to a metabolite, that can be variable. We have transporters on cells that can bring drugs into the cell and kick drugs out of a cell. And there's genetic polymorphisms associated with those transporters, so that can have some variability. And any sort of endogenous or exogenous inducers or inhibitors of either drug concentration or drug activity can also have an effect. So our lab, instead of focusing on the pharmacology of antiretrovirals in plasma, we spend our time focusing on the pharmacology of antiretrovirals at the site of action. And for this particular talk, it's the site of action for HIV prevention. Now, back in February of this year, during the State of the Union address, Donald Trump announced that he would like a plan for America to end the HIV epidemic. And his goal is to reduce HIV infections to 90%, or by 90% in 10 years. So that would put our infection rate at less than 3,000 a year, which would be below the definition of an epidemic. And this plan has 4 components to it. One is to diagnose all people with HIV very early. Number 2 is to treat people with HIV with antiretrovirals so that they have no viral replication going on in their plasma. To prevent new HIV infections by using pre-exposure prophylaxis, and to also respond quickly to any HIV outbreaks that happen so that we can get antiretrovirals to the people who need them for treatment, and then also for prevention. We are targeting a certain number of counties, 48 of the highest burden of HIV, along with Washington, D.C., San Juan, Puerto Rico, and 7 states that have a high rural burden of HIV. And so if you look at this map, this outlines our counties and also the states, and depending on how you define the Southeast, really the Southeast is kind of the ground zero for HIV infection. We have the highest rates of infection, so this becomes particularly important to us at UNC when we're thinking about serving the Southeast. So to end the epidemic, we need to stop HIV transmission, essentially. And so how do we stop transmission? We make the fluids that are infectious non-infectious. So the fluids that are infectious are blood, semen, rectal fluids, vaginal fluids, and also breast milk. And so because of that, HIV is transmitted through sex, as a result of pregnancy, childbirth, and breastfeeding, and through injection drug use. Occasionally through occupational exposure, through a needle stick, and very rarely through blood transfusion or organ transplant, because we have a lot of screening capabilities for that. So if we look back at these 4 topics to try and reduce the epidemic of HIV in the US, we're going to focus on two things. First is to treat people with HIV to make these bodily fluids non-infectious, and the other is to prevent new transmissions by using pre-exposure prophylaxis, which means we need to protect the sites that become infected with HIV. So that includes the vagina, the cervix, and colorectal tissue. So this is how I got into this area. I've been asked this a lot. So here I am as a young, enthusiastic assistant professor who's doing her own work focused on understanding and predicting drug interactions during the drug development process and other things. And Mike Cohen comes along, and Mike Cohen is the chief of infectious diseases in the School of Medicine at UNC at the time, also the director for the Institute for Global Health and Infectious Diseases, and the associate vice chancellor for global health. And so he says, you need to study antiretrovirals in semen, and you need to tell me how you're going to do this. Are we using dogs? Are we using monkeys? Are we using cats? Figure it out. You need to figure out how much drug is in semen. And so, me, not really being an animal person, was thinking, well, yeah, I don't really know how to do this or even why I'm doing this. We did do some work in cats because feline immunodeficiency virus is actually very similar to human immunodeficiency virus. But really, as a clinical pharmacologist, what this came down to was if we want to study drugs in semen, we have to study drugs in people. And so we did. So this was the foray into treatment as prevention. If you treat somebody with antiretrovirals, can you get them to such a place that they are no longer infectious to their partner? And so that's really what Mike was asking me to do. He said, I need to figure out what drugs give us the best chance of making HIV-infected individuals non-infectious so that there is no transmission. So we did a lot of work on drug concentrations in fluid that can transmit HIV, and we did start with semen, and I'm going to show you kind of a summary slide of these data. These data are presented as a ratio, so the concentration in semen divided by the concentration in plasma. So if you're looking at the line of unity, or 1 on this slide, anything that falls above that line means that the drugs are concentrating in semen. Anything that falls below that line means that the drugs are less concentrated in semen than they are in plasma. And these 5 categories are the 5 drug categories, therapeutic categories that we have for HIV infection. So here's what semen looks like. So for the most part, the drug concentrations tend to lie below the line of 1, although there is a set of drugs called nucleoside or nucleotide reverse transcriptase inhibitors that have concentrations that are higher than in plasma, which is a good thing. We also did the same thing in cervicovaginal fluid, so we can see generally that those concentrations tend to be a little bit higher than in semen. And then we also did colorectal fluid, and those concentrations tend to be even higher than they are in semen or in cervicovaginal fluid. So these provided us with clues to selecting those drugs to prevent HIV transmission. So we did a lot of other mathematical modeling work. We wanted to know how many doses of these drugs does it take to get to effective concentrations. And so we did that in semen, and the 2 drugs that I have here are zidovudine, which was the first drug that was available for HIV infection, and then lamivudine, which paired with zidovudine. And so we looked at drug dosing over time to see at what point drugs achieved high concentrations in the semen. Because these drugs also have to be phosphorylated inside the cell in order to be active against HIV, we also looked at cells within semen to see when those active phosphorylated metabolites would achieve highest exposures. And we also looked at how many days it took until HIV was undetectable in semen. And these are some data with another drug called Efavirenz, where in the small square we have plasma concentrations of HIV declining over time on treatment. And in this big square, we have HIV RNA in semen declining over time. So in general, it took about 40 days for HIV to no longer be found in semen, although we did have some discrepancies here where we had one patient who didn't respond in plasma but did respond in semen, and another patient who responded in plasma but did not respond as quickly to that drug in semen. So there are some caveats that we found to putting people on therapy and making them non-infectious. So this illustrates pharmacokinetics on the left side and then pharmacodynamics on the right side. And what we're talking about, again, is very simple and getting back to the basics of clinical pharmacology, which is getting the right drug to the right site in the right concentration for the right amount of time. So the data that we generated were used to select the drugs for this pivotal study called HPTN-052. So HPTN stands for HIV Prevention Trials Network, and it was a study that enrolled about 1,700 HIV serodiscordant couples. So one person was HIV infected and the other one was not. And these couples were split into two groups. In one group, the person who was HIV infected received antiretroviral therapy immediately. In the second group, that HIV-infected person received treatment in a delayed fashion until their CD4 count was at a certain level, according to guidelines, which told us when to treat HIV-infected people. The study was stopped early because we found the Data Safety Monitoring Board found a big difference in infection rate between those individuals who were treated early and those individuals who were not treated until later. So there was 1 infection in the immediate group, 27 infections in the delayed group. So the study was stopped early, people were still followed, and they found 2 more individuals in the early treatment arm who became infected. Now these 3 individuals altogether became infected either because they were not protected during the early part of antiretroviral therapy. So as I showed you, HIV takes a while to get to undetectable levels in semen, or the infected partner wasn't quite taking their drugs correctly. But this was the first definitive study that suggested that HIV treatment prevents HIV transmission. And so this became science's breakthrough of the year. And Mike Cohen said from the time the first AIDS drugs were developed in the mid-90s, our UNC team of virologists, pharmacologists, and physicians had been working on the idea that antiretrovirals might make people less contagious. And by 2000, the study team thought the idea was strong enough to try and prove it. So this is one way that our antiretroviral pharmacology work was able to select the right options to take into treatment to make people who are infected non-infectious. So if you've heard this phrase, U equals U, this comes from the treatment as prevention field, where undetectable, in this case undetectable HIV in plasma, means untransmittable. So now I'd like to move to pre-exposure prophylaxis. So that was treatment as prevention. So that was that second strategy in that 4-strategy approach for eliminating the epidemic in the US. Now that third strategy is pre-exposure prophylaxis, or PrEP. There are a lot of different forms of PrEP products being studied currently. I'm going to show you what we've done in the gel space and then also the pill space. But there are vaginal rings being developed, there are long-acting injectables, there are implants, there's all kinds of things that are currently being evaluated in terms of giving an antiretroviral to an uninfected individual to protect them from HIV acquisition. So tenofovir is one of these drugs. So tenofovir is a fairly potent antiretroviral used very commonly in HIV treatment. And it was decided to try and use this topically to prevent HIV infection. So tenofovir was formulated into a 1% gel, and it was used in a study involving 800 women in South Africa, and the study was called Caprisa 004. So they broke these women into 2 groups. It was a double-blinded, randomized, placebo-controlled trial. Half of the women got tenofovir gel, and the other half got placebo. And they were asked to use the gel up to 12 hours before sex, and then a second dose up to 12 hours after sex. And so what this study found was that the tenofovir gel provided a 39% lower HIV incidence than the placebo group. So it was effective, but it was sort of modestly effective. Regardless, This was still touted as a promising preventive. This was the first study where an antiretroviral was used to protect someone from HIV infection and definitely showed a significant effect. And even in Durban, South Africa, at the SeaWorld Aquarium, the tank divers in the Ray tank got very excited and had "Hooray for Tenofovir gel" as a banner in their tank. And Durban was where the study was initiated. There was another interesting thing that was found, and that was that the tenofovir gel also reduced herpes infection, or HSV2 infection. And so it reduced herpes infection by also 51%, which was sort of unexpected. Because that was unexpected, the whole study was put into question as to, is the gel really working? And is this the maximum effect we can expect out of the gel? And is the HSV2 effect real? And so we were at least able to collect in that study cervicovaginal fluid concentrations from women. You couldn't do tissues in this study because you couldn't do biopsies. A biopsy would put a woman at risk, increased risk, for HIV acquisition. So we looked at the fluid in the vaginal canal, and we binned people according to the concentration of tenofovir in the vaginal canal compared to the proportion of women who were infected with that concentration. And in fact, we saw a pretty good linear relationship between HIV protection and higher tenofovir concentrations in cervicovaginal fluid. So that was definitely a real effect. And if women had really high concentrations of tenofovir in the vaginal canal, they were completely protected from HIV. In terms of HSV2, it was a bit of a different sigmoid relationship, but still a positive relationship, where if you had higher concentrations in the vaginal canal, you could protect yourself from herpes. So this was an example of pharmacology at the end of a study, trying to go backwards and explain the results of a clinical trial, which we were able to do. Now, again, so this is the right drug to the right site and the right concentration for the right amount of time. And we determined that the results of the gel were fairly modest because it was very hard for women to use this product. 12 hours before sex, 12 hours after sex, how do you carry around the applicators? Everything was really messy. And so the women who used this gel as they were supposed to was a very small proportion of the total population. So while this is effective, it has no good utility in a population because it's very hard to use. So now you've probably seen this commercial, "I'm on the pill. It's not birth control. Find out if Truvada for PrEP may be right for you." So now let's take the tenofovir, that same compound that was in the gel. Let's add another compound to it called emtricitabine and put it in a fixed dose combination, one pill called Truvada. Now Truvada has, again, been used for HIV treatment, and it was being considered for use as HIV prevention as well. Now, there were a lot of mixed results in the early days with Truvada as PrEP, and some of them are here, and I want to focus on these two to start with. The IPREX study on the left, which was in MSM, men who have sex with men, and transgender women, so this was a male study, and then the FemPrEP study, which is on the right, which was focused on women in Africa. So the IPREX study had 44% efficacy, again, kind of modest efficacy for men who were given daily Truvada to try and protect themselves from HIV infection. Absolutely no effect in women in the FemPrEP study. In fact, it was stopped early, and the question was, well, does PrEP really work? Is it ready for the world? And then that next study to the right of FemPrEP was called VOICE, and even VOICE was stopped early. So there was debate as to whether Truvada works in women at all. So we were interested in this because the feeling at the time was that, well, if something is working for treatment, it should work for prevention, and we can just use the same thing in different populations. But we were very curious about whether this was getting the right drug to the right site in the right concentration for the right amount of time. So we're going to take a deep dive into this particular approach so that I could show you how pharmacology principles are able to inform public health. So let's look at the early events in HIV infection. So you can see red HIV particles here, which are crossing an epithelial barrier, either the vagina, the cervix, or colorectal tissue. You may have a dendritic cell here that's picking up some virus, but really where it's headed is to the activated CD4 T cells that are in mucosal tissue, and we have them in all different types of mucosal tissue. This is sort of ground zero for viral infection, and in all animal studies that we've seen, the infection always starts in mucosal tissue. Eventually, over days, you have virus moving to regional lymph nodes and setting up shop there, and then the virus is spreading to the rest of the body. There is some suggestion that dendritic cells may just traffic the virus right to lymphoid tissue, but that's more theoretical than we've actually had data on. So we really focus on the stroma or the lamina propria of mucosal tissue as where we need to abort infection. So the next question is, well, what is the target concentration that we need to abort infection so that we can work backwards in terms of what kind of dosing strategy we need? Well, so if we go back to the right concentration and we just look at what we know about the female genital tract in pink and colorectal tissue in blue, we can see that generally concentrations in the female genital tract are actually quite a bit lower for antiretrovirals than concentrations in colorectal tissue. And if we look specifically at tenofovir, which is TFV, and emtricitabine, which is FTC, we see pretty significant differences in just tenofovir concentrations between female genital tract and colorectal tissue, just in fluid. So we figured we needed to really look at this in tissues. But before we did, we needed to understand how these drugs are working. So the drugs are administered orally, they get into the plasma, and then they have to cross cell membranes and get into the cell. The active form of the drug is the phosphorylated form of the drug. So tenofovir needs to be phosphorylated twice to a tenofovir diphosphate. FTC needs to be phosphorylated 3 times to FTC triphosphate in order to block HIV replication. So we know we need to be looking at tenofovir diphosphate and FTC triphosphate in colorectal tissue, not just parent drug, or in any tissue, not just parent drug. And there's another piece that we need to look at, because the efficacy of these active metabolites is a direct result of the amount of competing endogenous nucleotide that is in the cell. So what we have here is HIV RNA that is now being transcribed into HIV DNA. So HIV is an RNA virus that uncoats, and then RNA is transcribed into DNA, and then that DNA is incorporated into host cell genetic material. So during this chain elongation of DNA, typically you have nucleotides coming in and depositing themselves and reading the RNA. So the way these drugs work is they are nucleotide analogs, and they fall into this chain, and they are chain terminators, so you can't add anything onto these drugs. So DATP is the competitor for tenofovir diphosphate, and DCTP is the competitor for FTC triphosphate. And based on some data from the CDC, they've been able to show that as you increase the endogenous nucleotide, you decrease the efficacy of these drugs. So not only do we need to look at the active forms of the drugs in the tissue, we need to look at the nucleoside analogs in those tissues as well. So we've got some complicated pharmacology that we're doing, but bear with me. So what we did is we took just a single dose and gave it to a number of women and men, and we looked at the tenofovir diphosphate and the FTC triphosphate concentrations in the female genital tract, in those tissues, and also in colorectal tissue. And we saw something interesting. So for tenofovir diphosphate, rectal concentrations are in blue, and female genital tract concentrations are in pink. So what we have for tenofovir diphosphate is a very high extended exposure in colorectal tissue, but not in the female genital tract, which is similar to what we saw for fluids. For FTC triphosphate, we actually see quite high concentrations in the female genital tract initially compared to colorectal tissue, but those fall off really quickly. By 3 days, those concentrations are gone. So we have differences in two drugs that are in the same drug class, one that concentrates in female genital tract and one that concentrates in colorectal tissue. When we looked at the endogenous nucleotides that were also in these tissues, we were kind of surprised to also find differences. And in fact, we saw lower endogenous nucleotide concentrations in rectal tissue than we did in the female genital tract, so a lot more endogenous nucleotides in women. So then we decided, in order to combine these two pieces together, we looked at the ratio of the active form of the drug to its endogenous nucleotide. And we did some modeling to see at what point those concentrations would level out after multiple dosing. And so for the tenofovir diphosphate to DATP ratio, we saw, again, colorectal concentrations that were much higher, or ratios that were much higher than the female genital tract. So the ratio in the female genital tract was 0.5, and in rectal tissue it was 17, so a big discrepancy there. Whereas for FTC triphosphate, those ratios were about the same at the end. So even though the female genital tract had much higher FTC triphosphate concentrations, it also had much higher DCTP concentrations. So those sort of balanced themselves out. So now the question is, well, what is the target ratio for these tissues that would provide protection against HIV? So we couldn't do this in people because we can't give them different dosing strategies and in fact try and expose them to HIV and figure out, which is the best ratio to achieve. So we did this in cells. We did this in primary cells and peripheral blood mononuclear cells, and we evaluated Emax models for both tenofovir and emtricitabine, both alone and also in combination. So we did some surface modeling to understand, are these drugs synergistic? Are they additive? We found that they were synergistic, and we were able to model what sort of target concentrations or target ratios were needed to prevent HIV acquisition. And so these were our final results. So we did some simulation studies in populations, and we looked at the number of doses for these drugs individually and in combination per week that would result in protection of a person against HIV infection. And there are some big differences here. So in colorectal tissue, whether you're taking 1, 2, 3, 4, 5, 6, or 7 doses a week, so 7 doses a week is a daily dose. For emtricitabine, you do have a good proportion of the population that achieves that efficacy target, although it's not 100%. For tenofovir, because those ratios are so high in colorectal tissue, you don't need a whole lot of doses to protect colorectal tissue with tenofovir, and then when you use the combination, small dosing strategies lead to efficacy. But if you look at the female genital tract, we have very different results, where emtricitabine, maybe 50% of the population achieves that efficacy target. Almost none of the population achieves it with tenofovir, and then when you combine them in a synergistic interaction, you have about 85% of the population, if you take a dose every single day, achieving that target ratio. So now let's look back at these two studies, which was IPREX, the study in MSM and transgender women, and also the FemPrep study for women in Africa. So the investigators in the IPREX study did some modeling of their data, and they demonstrated that if someone was taking 2 doses of Truvada a week, they decreased their infection rate by 76%. If they were taking 4 doses a week, by 96%, 7 doses a week by 99%. Whereas if we look at the FemPrep study, this is monthly data of women over the course of a year who either had poor adherence in red, so less than 2 doses a week, modest adherence, 2 to 3 doses a week in yellow, or good adherence, 4 or more doses a week in green. And overall, 40 to 60% of these women were actually taking 2 or more doses of Truvada. Well, if you look at the data in men, 2 or more doses of Truvada should be providing protection, but it didn't because of the different distribution characteristics of these drugs in colorectal tissue compared to the female genital tract. And when a study was done called Partners PrEP, they found that if women were taking 7 doses of Truvada per week, they decreased their HIV incidence by 90%, and we had about 85%, so we were pretty close. So the modeling exercise that we did could actually predict what happened in the clinical trials, and had we done the pharmacology work up front, we probably would have saved a lot of grief in the clinical trials down the road. But the bottom line is, for Truvada as oral PrEP, it works. It's almost 100% effective when you take it as prescribed, and it's prescribed as a daily dose. Especially for women, this is really important, whereas men can get away with less. Now, there's a problem with daily adherence to Truvada. There are some side effects associated with it. You can feel a little nauseous, you can have an upset stomach, so now you're taking a healthy person with no symptoms, and you're giving them symptoms. Truvada can also cause a decrease in kidney function and a decrease in bone density over time, so that needs to be followed. There's also a stigma associated with carrying around pills. You can be perceived as HIV positive. You may be accused of not trusting your partner. There's also a lot of complexities around people's daily life and their priorities, like housing and employment and food security and mental health issues. These can all get in the way of taking a daily dose of a pill. And there are also seasons of risk. So you may be at risk one weekend, and you're not at risk for acquiring HIV for another month. But people are being asked to take this drug on a daily basis. So one last study I want to show you is IPERGAY, which was a study in MSM that was done in France and in Canada. And it also had a very good efficacy rate of 86%. This was to try and get at intermittent dosing. Do you really need to take a dose every single day, or can this be coitally dependent or event-driven PrEP? So these men were asked to take 2 tablets of Truvada up to 24 hours before sex, 1 tablet 24 hours after sex, and then another tablet 48 hours after that. So you ended up taking 4 doses around the time of exposure. And what they found was pretty significant, where there were only 2 infections in the Truvada arm and 14 infections in the placebo arm, and so the participants were unrandomized and they were all offered PrEP. Now, if you look at their adherence by pill count, if they had 1 event a week, then they should be taking 16 doses a month. But when you look at how many doses people were taking up to 30 doses a month, which is essentially daily dosing. So there was some pretty significant use of Truvada in this population. It just wasn't, one and done for a month and then one and done again. So one of the criticisms of this study as well is, can you really do this coitally dependent, or did you have such low rates of infection because these guys were having a lot of sex and taking a lot of pills? So we decided to do some modeling with just single exposures. And I just want you to focus on these dotted lines. We looked in colorectal tissue on the left, and we looked at female genital tract tissue on the right. And so this is the time of coitus, and we have some drug dosing before and after that. But then as you look over time, 3 days after the last dose, the concentrations or the proportion of people who are achieving target is really high for colorectal tissue, but it's dropping off in the female genital tract. If you look at 5 days after the last dose, again, high numbers of people who still are achieving that target of efficacy in colorectal tissue, even less in the female genital tract. And then by 7 days, you have, again, high exposures in colorectal tissue and almost nothing in the female genital tract. Now, we don't know how long we need to use antiretrovirals to protect against infection. At least in vitro systems, it's at least 8 days. So I think we need at least coverage for 10 days. So doing this to protect colorectal tissue is probably totally fine. Doing it to protect female genital tract tissue is not a good idea because those concentrations drop off pretty quickly. So in our hands, we've recommended that if someone wants to use a coitally dependent dosing strategy, it's okay if you're protecting colorectal tissue. It's not okay if you're protecting the female genital tract. You're going to have too many infections. So the lessons learned for us was that antiretroviral treatment prevents transmission of HIV, and PrEP prevents acquisition of HIV. The PK/PD analysis that we did for treatment as prevention was really critical to optimizing therapy, and any clinical results that we saw with the PrEP studies would have been very difficult to explain without the pharmacology data. We know that antiretroviral distribution in mucosal tissues is heterogeneous, so if you try to use plasma data to extrapolate efficacy of Truvada, men and women have very similar plasma concentrations, but they have very different tissue concentrations, so that would have been inadequate for us. And understanding some of the cellular biology was also helpful to provide insights into PrEP efficacy. And so we can do a pretty good job at predicting how clinical trials will turn out if we have some PK data up front in people, and we can do some in vitro investigations in terms of efficacy targets. So we can determine what sort of PrEP dosing strategies we should have, evaluate how forgiving a regimen is if you miss a dose, how critical is that to infection, and we can explain clinical trial results. However, the PrEP challenges remain, and even the antiretroviral treatment challenges remain in terms of linkage and retention and care. So if we just look at PrEP, right now we have 1.2 million Americans that are estimated to be at high risk for HIV infection. Less than 10% of them use PrEP. So we have effective tools, but we have a long way to go in terms of implementation and uptake. So with that, I'd like to acknowledge a number of collaborators, both within and outside of UNC, all of our funding sources, and I'll leave you with this picture of the lab, which has a Nelson Mandela quote in front of it. It always seems impossible until it's done, and I think my lab lives by that every day. So thank you so much for your attention.
Do you think that there could ever be something where with the birth control pill, there's also Truvada built inside of that, so you're taking both at the same time?
Yeah, what a great question. So yes, and so people are working on that now. This is called a MultiPurpose Technology because it does multiple things with one pill or one vaginal ring or one injection. Through the studies of prevention in women, it's become apparent that women are more worried about falling pregnant than they are about acquiring HIV. So their primary motivation is contraception and not protecting themselves from HIV, because most of the time, researchers identify people as being at risk for HIV, but people don't identify themselves that way. And so that has been the next wave in prevention efforts, that we don't have a product on the market yet, but it's being investigated. So let's give women something for contraception that they want to use, however it comes, as a pill, as a vaginal ring, as an injection, and say, oh, by the way, we're going to add some HIV protection on top of that. And maybe we'll add some herpes protection on top of that. So we can use these technologies for multiple purposes. So it's a great question, and it's being explored now.
It seems that, I'm very tall. Seems that men kind of got lucky with how well their bodies are able to retain the medication in the majority of the studies that you cited there. So do you know if there's any other avenues they're looking for women that's not via Truvada that could, just like seeing if they would also be able to find a drug that women would be able to maintain in their bodies as well, so it's not so dependent on taking a dose every single day.
Right. So the one thing is a great question. The one thing that's being investigated in that space is long-acting injectables. So there's another drug that Glaxo or ViiV Healthcare has that's called cabotegravir. And cabotegravir, they've been able to mill into a fine nanoparticle like suspension, and they give 2 injections in the butt, 2 mil injections, 1 in each butt cheek, and that lasts for about 2 months. The data on how effective it is in prevention is still being collected. There's less of a discrepancy between female genital tract tissue and colorectal tissue with this formulation than there is with Truvada. So it might be that we'll have something that does get closer and allows women as much protection as men with the same dosing strategy. Now, when we've looked at some of the data in monkeys, it does appear that there's slightly lower concentrations in the female genital tract than there is in colorectal tissue in monkeys, but that difference is a lot smaller than what we see with Truvada. So stay tuned on that. Those data are still being collected.
Hi. So you have a lot of data that supports that, these drugs affect the sexes differently. How would someone who is trans that uses hormone therapy, how does that work and how would that affect them?
Right. No one has investigated that question up to now. So I have an investigator in my laboratory, her name is Mackenzie Cottrell, who has an NIH grant to look at this specifically, because the trans population, whether it was men or women, were just getting the same drug as everybody else without any consideration for what the hormones are doing and what that looks like in mucosal tissue. So she has just enrolled a cohort of trans men, and she's going to be working on a cohort of trans women to be able to evaluate who are on PrEP, standard doses of PrEP, of Truvada, to evaluate what those concentrations look like and what the hormone levels are and whether we need to worry about that population in terms of getting that dose right. So the study has been done, the data are collected, they're being run in the lab this week, so we'll see. It's a great question.
Hi. So your bio is super long and super impressive, but I guess my question is, what's the most fulfilling part of your job? The research, the teaching, the speaking, like what?
Yes.
Like, I guess just what's most fulfilling?
Yeah.
Like,
So what gets me up every morning and makes me tap dance to work are a couple things. One is trying to optimize therapy for the people that need it, and to make sure that clinicians have the tools they need to be able to prescribe therapy most appropriately. So we're always working on clinical dilemmas and applying that right back into patient care. So that part is very fulfilling, problem-solving clinical dilemmas. But the other part that's very fulfilling is working with a wonderful group of investigators in my lab and a wonderful group of trainees, seeing them develop their ideas, seeing them develop data and generating data and growing scientifically is exceptionally rewarding.
Hello. You said that about only 10% of people labeled as risk for contracting HIV are under PrEP medication.
Yeah.
And do you think that the majority or the reason behind this is lack of information, lack of access, both, or something else?
Yeah, I think it is a combination of things. So part of it is lack of information, although a number of groups have been trying very hard to get the word out that PrEP works, and people should be on PrEP, people should be at least asking for PrEP. But then there is access issues. People going to a family physician, the family physician may not totally understand PrEP, may not want to prescribe PrEP, so we need to make sure that there are safe places for people to talk about sexual activity and what their risk is, and have access to that medication. Cost is also another issue. We're hoping that that's going to be eliminated with the President's strategy that everybody who needs it will get it, regardless of what sort of insurance they have or if they don't have insurance. So there are a lot of issues. And then some people, again, this is kind of a daily dosing strategy, and people don't want, as a healthy person, they don't want to be taking something that kind of makes them feel sort of icky or that they don't need consistently. So once we have more options that are coming down the pike, I think that will also help, because everybody doesn't want to take a pill a day. It's a good question. So a lot of hurdles to overcome for sure.
So a lot of this focuses on MSM, so men having sex with men, or and that was colorectal tissues mostly, and then you had vaginal tissues mostly. What about like women having sex with men, other categories of transmission?
Right. So yeah, so transmission through foreskin, for example, right. We are not exactly clear on what those concentrations look like for PrEP. We tried to do a study once with Gilead, and then they kind of put the kibosh on it and didn't really want us to look at that. So we don't have any data on that. So we have to make some assumptions about what those concentrations look like. And that may be a reason why we don't see necessarily 100% efficacy in some of the studies, but we see at least 90% efficacy. So it's a good question, and we don't exactly know what those concentrations look like.
So what do you think is the next big thing in PrEP? Like, what's the most exciting thing that's coming out?
So I think so cabotegravir is coming soon. So the long-acting injectable, that's not that long-acting because you have to keep coming back every couple months. So that is a bit concerning. So that will be next up, and we'll see what that looks like. The multi-purpose technologies are pretty exciting. We now have some new antiretroviral compounds that we think we can put as an implantable, so something under the skin that can stay there for a year. So it may be that you need an implantable and you're good to go over the course of a year. So I think those in particular, if you're combining it with contraception, for example, or other things, could be particularly attractive to people. So they don't need to think about it on a daily basis or a monthly basis. It provides multiple forms of protection and can keep people safe for extended periods of time. I think I've exhausted them. Shall I dismiss? Thank you so much. It was a pleasure to be here.